So here's something worth thinking about as you settle in tonight. Right now, inside your skull, roughly 86 billion neurons are doing something extraordinary. They're talking to each other. Not with words, obviously, but with tiny electrical impulses, little bursts of voltage that ripple across the surface of your brain in waves. Actual waves, like waves on water, except these are waves of electricity, and they've been rolling through your head every single moment of your life, waking and sleeping, since before you were born. And the wild thing is, for most of human history, nobody had any idea they were there. No one even suspected. You could hold a human brain in your hands, study it, dissect it, write entire medical textbooks about it, and you would never once notice that the whole thing was humming with electrical rhythms. Because you can't see them. You can't feel them. You can't hear them. They're invisible, silent, and incredibly faint. We're talking about voltages so small, they're measured in millionths of a volt. To put that in perspective, the static shock you get touching a doorknob in winter is something like 25,000 volts. The electrical signal coming off your brain is maybe 10 or 20 microvolts. That's 1,000,000 times smaller. It's almost nothing. And yet those tiny, almost nothing signals turn out to be one of the most important things happening inside you right now. But let's not get ahead of ourselves. Let's start where this story actually starts, which is in a pretty unlikely place. A physiology lab in a small German university town in the 1920s, with a man who spent nearly 20 years trying to prove something that most of his colleagues thought was, frankly, a little bit nuts. His name was Hans Berger, and if you've never heard of him, that's sort of fitting, because for most of his career, almost nobody took him seriously. Berger was born in 1873 in Neuses, a village in Bavaria, and by all accounts, he was a quiet, meticulous, somewhat obsessive person, the kind of scientist who kept detailed diaries of every experiment he ran, who repeated measurements over and over and over before he'd trust them. He studied medicine, became a psychiatrist, and eventually landed at the University of Jena, which was a perfectly respectable institution, but not exactly the center of the scientific world. And starting around 1902, Berger became consumed with an idea that would dominate the rest of his life. He wanted to find a way to measure the energy of the human brain. Now, here's the thing about Berger. His motivation wasn't purely scientific, at least not at first. There's a story, and it's one he told himself, so we have reason to believe it. When he was a young man doing his military service, he had a riding accident. He was thrown from his horse directly into the path of a horse-drawn cannon, and he very nearly died. That same evening, his sister, who was many miles away, suddenly felt an overwhelming sense of dread, a conviction that something terrible had happened to Hans. She persuaded their father to send a telegram asking if he was all right. Berger was deeply shaken by this. He became convinced that in the moment of his near-death experience, his brain had somehow transmitted energy, some kind of psychic signal that his sister had received. And he wanted to find that energy. He wanted to measure it. Now, I know what you might be thinking. That sounds less like serious neuroscience and more like the beginning of a ghost hunting show. And honestly, his colleagues thought something similar. But here's what's fascinating about Berger's story. He was wrong about the telepathy, completely wrong. But in chasing that wrong idea, he stumbled onto something real and genuinely revolutionary. Because to look for psychic energy coming from the brain, he first had to figure out whether the brain produced any measurable electrical activity at all, and that question, it turns out, was a very good one. Berger wasn't working in a vacuum, of course. By the time he started his experiments, scientists already knew that animal brains produced electricity. A British physician named Richard Caton had demonstrated this back in 1875, placing electrodes directly on the exposed brains of rabbits and monkeys and detecting faint electrical currents with a very sensitive galvanometer. Basically, a device that measures tiny amounts of electrical flow. Caton noticed something interesting. The electrical activity changed depending on what the animal was doing. When the animal was alert and looking at something, the signal shifted. When it was resting, they shifted again. Caton published his findings, and they were mostly ignored. Partly because the measurements were so faint and so hard to reproduce, and partly because nobody really knew what to do with the information. What did it mean that the brain was electrical? How did that connect to thought or consciousness or anything useful? Nobody had a framework for thinking about it. So when Berger started trying to record electrical signals from the human brain, from the outside through the intact skull, most people thought he was wasting his time. The signals, if they existed at all, would be absurdly weak. They'd be buried under noise from the muscles of the scalp, from the heartbeat, from the electrical equipment in the room. It would be like trying to hear someone whisper from across a football stadium during a rock concert. And Berger didn't have fancy equipment. He started with a simple string galvanometer, the same basic technology Caton had used decades earlier, and he spent years getting nowhere. But he kept going. This is one of those things about the history of science that I think is really worth sitting with for a moment. Berger worked on this problem, largely alone, for close to 20 years before he got results he trusted enough to publish. 20 years. Think about what that means. Year after year, going into his lab, placing electrodes on patients' scalps, sometimes his own son's scalp, fiddling with equipment, getting noisy, ambiguous readings, writing it all down in his meticulous diaries, and trying again. His colleagues at Gina knew he was doing something in his lab, but he was so secretive about it that most of them had no idea what. He was terrified of being ridiculed. He was terrified of being wrong. And then, sometime around 1924, things started to click. Berger had gotten his hands on a more sensitive recording device, a Siemens double-coil galvanometer, and he'd refined his electrode placement technique. He was using small silver foil pads placed on the forehead and the back of the head, connected by wires to his galvanometer. And on July 6, 1924, he made his first successful recording of electrical activity from the human brain, through the intact skull. He recorded it from a 17-year-old patient named Zedel, who was undergoing a neurosurgical procedure. The young man had a piece of skull temporarily removed, which gave Berger a chance to place electrodes closer to the brain itself. And there it was, a rhythmic, oscillating signal, a wave. Berger didn't publish this result for another 5 years. 5 more years of checking, rechecking, recording from more subjects, trying different electrode placements, ruling out artifacts. Was the signal really coming from the brain? Or was it just picking up the heartbeat, or muscle twitches, or electrical interference from the building? He tested all of these possibilities, painstakingly, one by one. He recorded from himself. He recorded from his son Klaus. He recorded from patients with skull defects, where the electrodes could get closer to the brain tissue. And gradually, he built up an airtight case. In 1929, Hans Berger published a paper titled Über das Elektrencephalogramm des Menschen, on the electroencephalogram of man. It was the first of 14 papers he would publish on the topic, and in it, he introduced the word that we still use today, electroencephalogram, EEG, literally, a writing of the electrical activity of the brain. And in that paper, he described two distinct types of brain waves that he'd observed. The first, and most prominent, was a rhythmic oscillation that appeared when the subject was relaxed and had their eyes closed. It cycled at a frequency of about 8 to 12 times per second, 8 to 12 hertz in the units we use for frequency. Berger called this the alpha wave, and the second type was faster, smaller, and appeared when the subject opened their eyes or started concentrating on something. He called this the beta wave. Let's slow down here for a second, because these two observations, alpha and beta, are really the foundation of everything else we're going to talk about tonight, and they're worth understanding clearly. When Berger said he was seeing a wave at 8 to 12 hertz, what he meant was this. The voltage being picked up by his electrodes was going up and down in a regular rhythm, completing somewhere between 8 and 12 full cycles every second. If you've ever watched the ocean and counted waves passing a fixed point, say 1 wave every 5 seconds, you've done exactly this kind of frequency measurement. Berger was counting brain waves, and the alpha rhythm was rolling through at a pace of roughly 10 per second. Gentle, steady, rhythmic. And here's what was so striking about it. The alpha wave was most prominent when the person wasn't doing anything in particular. Eyes closed, relaxed, not trying to solve a math problem or read a book or focus on anything specific. Just resting. The moment the person opened their eyes or started concentrating, the alpha would diminish or disappear, replaced by the faster, lower amplitude beta activity. Berger called this the alpha blocking response, and it was one of the first clues that brainwaves weren't just random noise. They were functionally meaningful. They changed depending on what the brain was doing. The brain's electrical rhythm was somehow linked to its state of consciousness. Now, you might think that the scientific world greeted this discovery with excitement and celebration. It did not. Berger's 1929 paper was met with almost total indifference, and in some quarters, outright skepticism. Part of this was the telepathy thing. Berger's earlier interest in psychic phenomena had given him a reputation as a bit of a crank, and some scientists simply didn't trust his results. Part of it was that the signals were so incredibly small that many researchers doubted they were real. And part of it was just the conservatism of the scientific establishment. The idea that you could stick electrodes on someone's scalp and read meaningful information about what their brain was doing, it sounded too good to be true, too simple, too weird. The person who changed everything was a British physiologist named Edgar Adrian. Adrian was, in almost every way, the opposite of Berger. Where Berger was obscure and self-doubting, Adrian was one of the most respected scientists in England. He'd won the Nobel Prize in Physiology or Medicine in 1932 for his work on the function of neurons. He was based at Cambridge. He had credibility to burn. And in 1934, Adrian decided to try to replicate Berger's results. He set up his own EEG recordings, using better equipment than Berger had access to, and he confirmed the findings. The alpha rhythm was real. It was coming from the brain, and it behaved exactly the way Berger said it did, present during relaxation, suppressed during attention. Adrian presented his results at a meeting of the Physiological Society in England, and suddenly, everything changed. When Edgar Adrian said the alpha wave was real, people listened. Almost overnight, Berger went from being dismissed as a fringe figure to being recognized as a pioneer. Laboratories across Europe and America started building their own EEG machines. The field of electroencephalography was born. And this is where the story really opens up, because once scientists had a tool for watching the brain's electrical activity in real time, they started noticing things that nobody had expected. The brain wasn't just producing alpha and beta waves. It was producing a whole symphony of different rhythms at different frequencies in different brain regions, changing constantly depending on what the person was doing, feeling, or experiencing. And each of these rhythms, it would turn out, was telling a different part of the story of what it means to be a conscious, thinking, sleeping, dreaming human being. One of the first and most dramatic discoveries came from studying something you're probably getting closer to right now: sleep. Because when researchers started recording EEGs from people as they fell asleep, they noticed something remarkable. The brain waves didn't just slow down, they transformed. The patterns changed in ways that were so distinct, so consistent from person to person, that you could literally watch someone's EEG trace and tell exactly when they crossed the threshold from waking to sleeping. And as the night went on, the waves kept changing, cycling through patterns that nobody had imagined existed before EEG made them visible. And this is one of those moments in science where a tool doesn't just answer a question. It reveals that the question was far bigger than anyone realized. So let's stay with this for a while, because the story of how scientists mapped the sleeping brain is genuinely one of the most fascinating chapters in all of neuroscience, and it connects directly to what's happening inside your head right now, tonight, as you lie here listening. The key figure in this part of the story is a man named Alfred Lee Loomis. And Loomis is one of those people history doesn't talk about nearly enough. He wasn't a neuroscientist. He wasn't even a full-time academic. Loomis was a Wall Street lawyer and financier who had made a fortune in the 1920s, gotten out of the stock market just before the 1929 crash with almost uncanny timing, and then essentially retired from finance to pursue his real passion, which was science. He built a private laboratory in a mansion in Tuxedo Park, New York, and he filled it with the best equipment money could buy. He invited top scientists to come work there. He funded research out of his own pocket. And in the mid-1930s, Loomis became fascinated by EEG and decided he wanted to study sleep. Now, here's what made Loomis's approach different. Most EEG researchers at the time were recording brainwaves for a few minutes at a stretch, maybe an hour. Loomis wanted to record entire nights. He set up bedrooms in his laboratory, invited volunteers to sleep there, and ran continuous EEG recordings from the moment they closed their eyes until they woke up in the morning. This was in 1937, just a few years after Adrian had validated Berger's work. And what Loomis and his colleagues found was extraordinary. They discovered that sleep wasn't one thing. It wasn't just the brain going quiet or slowing down uniformly. Instead, the brain cycled through distinct stages, each with its own characteristic electrical signature. Loomis identified 5 stages, which he labeled A through E, and each one looked different on the EEG trace. Stage A was drowsiness. You'd see the alpha rhythm start to break apart, becoming less regular, interspersed with slower waves. Stage B was light sleep, where the alpha rhythm disappeared entirely and was replaced by lower voltage, mixed frequency activity. And then things got really interesting. In stage C, Loomis noticed something peculiar. Short bursts of rhythmic activity would appear. These quick little spindle-shaped clusters of waves, lasting maybe half a second to a second, oscillating at around 12 to 14 cycles per second. He called them sleep spindles, and that name has stuck ever since. They looked like someone had briefly turned on a little oscillator in the brain, and then turned it off again. These spindles seemed to mark the transition into genuine, committed sleep, the point where you'd stopped just dozing and had actually fallen properly asleep. And if you think about your own experience of falling asleep, there's often that moment where you're drifting, kind of half aware, and then suddenly you're just gone. That transition, that crossing of the threshold, corresponds remarkably well to the appearance of sleep spindles on an EEG. Stage D brought even slower, larger waves, what we now call delta waves. These are the slowest brain waves we typically see, oscillating at roughly half a cycle to about 4 cycles per second. But they're big. The voltage is much higher than what you see during waking. Think about what that means physically. During deep sleep, huge populations of neurons are firing together in slow, synchronized pulses. Millions of cells, rising and falling in electrical activity in unison, like a stadium crowd doing a slow-motion wave. That's delta activity, and stage E, the deepest stage Loomis identified, was dominated almost entirely by these large, slow delta waves. So within just a few years of Berger's work being taken seriously, researchers had already discovered that the sleeping brain has its own complex internal structure. Sleep isn't unconsciousness. It's a different kind of consciousness with its own rhythms, its own stages, its own architecture. And here's what's really beautiful about it. These stages don't just happen once per night. The brain cycles through them repeatedly, in roughly 90-minute cycles, rising from deep sleep, back up toward lighter stages, and then descending again. You go through 4, maybe 5 of these cycles every single night. You're doing it right now, or you will be soon. But Loomis's classification, as groundbreaking as it was missing something huge, and it took another 15 years for someone to find it. In 1953, a graduate student at the University of Chicago named Eugene Oscillations made a discovery that would reshape our entire understanding of sleep. Oscillations was working in the laboratory of Nathaniel Kleitman, who was already one of the foremost sleep researchers in the world. Kleitman had literally written the book on sleep, a massive encyclopedic volume called Sleep and Wakefulness, published in 1939. He was methodical, rigorous, and deeply committed to understanding sleep as a physiological process rather than just the absence of waking. Oscillations' job was to study eye movements during sleep, particularly in infants. And one night, while monitoring a sleeping subject, he noticed something strange on his recording equipment. Periodically throughout the night, the sleeper's eyes would begin moving rapidly beneath their closed eyelids. Not the slow, rolling eye movements you sometimes see as people drift off, but quick, darting movements, back and forth, up and down, almost as if the person were watching something happen with their eyes closed. And when Oscillations looked at the EEG trace during these periods of rapid eye movement, he got a shock. The brain waves didn't look like sleep at all. They looked like waking. The trace showed low voltage, fast, desynchronized activity, almost indistinguishable from the EEG of someone who was awake and alert. Think about how strange that is. Here's a person who is clearly asleep. They're lying still. They're hard to wake up. Their muscles are relaxed. But their brain is producing electrical patterns that look like active, engaged wakefulness. Oscillations initially thought his equipment was broken. He checked the connections, he recalibrated the machine, he ran the recordings again. Same result. Every subject, every night, showed these periodic bursts of rapid eye movement accompanied by wake-like brain activity. Oscillations and Kleitman published their findings in 1953 in the journal Science, and they had discovered what we now call REM sleep, rapid eye movement sleep. And here's the kicker. When researchers woke people up during REM periods and asked them what was happening, the answer was almost always the same. They were dreaming. Vivid, narrative, emotionally intense dreams. Wake someone during non-REM sleep, and they might report vague thoughts or fragments, but wake them during REM, and you'd get full stories. Flying, falling, running, conversations with people who weren't there. So suddenly, the EEG had revealed something profound. Dreaming wasn't random. It wasn't scattered throughout the night. It was concentrated in these specific periods that had their own distinct neurophysiological signature. The brain was essentially switching into a completely different mode. A mode where it was internally as active as waking, but completely disconnected from the outside world. Your muscles are actually paralyzed during REM sleep. There's an active mechanism in the brainstem that suppresses motor output so you don't act out your dreams. The technical term is muscle atonia. Your brain is generating commands to move, to run, to speak, but those commands are being blocked at the level of the spinal cord. It's an extraordinary piece of biological engineering when you think about it. The brain has built in a safety system so that it can simulate experiences internally without the body actually responding. And this discovery, REM sleep, forced scientists to completely rethink their classification of sleep stages. It wasn't just a smooth gradient from light to deep. There were two fundamentally different kinds of sleep: non-REM sleep, which included the progressively deeper stages Loomis had identified, and REM sleep, which was something else entirely. Over the following decade, William Dement, another student of Kleitman's, did much of the painstaking work of mapping out the full architecture of a normal night's sleep. Dement would go on to become one of the most influential sleep researchers in history, eventually founding the Stanford Sleep Research Center and spending decades arguing, sometimes almost desperately, that society was dangerously undervaluing sleep. But let's come back to the brain waves themselves, because the taxonomy was growing. You had alpha waves in the 8 to 12 hertz range, showing up during relaxed wakefulness. Beta waves above that, roughly 13 to 30 hertz, associated with active thinking and concentration. Delta waves down at the bottom, half a hertz to 4 hertz, dominating deep sleep. Sleep spindles in that 12 to 14 hertz range, marking the onset of sleep. And then there was another rhythm that researchers started paying attention to, one that occupied the space between delta and alpha, a rhythm in the 4 to 8 hertz range that they called theta. Theta waves turned out to be particularly interesting because they showed up in several different contexts. You'd see them during drowsiness, in that hypnagogic state between waking and sleeping, that floaty, half-dreaming phase where thoughts start to become untethered from logic and images drift through your mind unbidden. You'd see theta activity during REM sleep, and intriguingly, you'd also see theta waves during certain kinds of waking tasks, particularly tasks involving memory encoding, navigation, and emotional processing. In animal studies, especially in rats, theta rhythms in the hippocampus, that seahorse-shaped structure deep in the temporal lobe that's crucial for forming new memories, turned out to be one of the most prominent and important electrical signals in the entire brain. So now, the picture was getting richer. The brain wasn't just producing one or two frequencies. It was producing an entire spectrum of oscillations, and different frequencies seemed to be associated with different functional states. Alpha for relaxed awareness, beta for focused attention, theta for memory and that twilight zone between waking and sleeping, delta for the deepest restorative sleep. And each of these rhythms wasn't just a passive indicator, like a thermometer reading. Researchers were beginning to suspect that the rhythms themselves were doing something, that they were functional, that they played active roles in how the brain processed information, consolidated memories, and maintained consciousness. But understanding what these rhythms actually do, mechanistically, that was a much harder question, and answering it required a completely different kind of investigation, one that would take researchers deep into the cellular architecture of the brain itself. Because if you want to understand why neurons oscillate, you have to understand how individual neurons work, how they talk to each other, and how the properties of neural circuits give rise to these collective rhythms that you can pick up on the surface of the scalp. And that story starts with a structure you might not expect, a small, walnut-shaped cluster of neurons buried deep in the center of the brain, called the thalamus, which turns out to be one of the most important rhythm generators in your entire nervous system. The thalamus sits right at the crossroads of nearly every sensory pathway in your brain. Almost everything you see, hear, and feel, visual signals from your eyes, auditory signals from your ears, touch information from your skin, all of it passes through the thalamus on its way to the cortex. Think of it like a relay station, a central switchboard. And for a long time, that's basically how neuroscientists described it. Just a relay, a pass-through, a place where signals got handed off from one part of the brain to another. But that turned out to be a massive underestimation. The thalamus isn't just passing signals along. It's actively shaping them, filtering them, and, here's the key part, it's generating rhythms of its own that profoundly influence what the cortex does. The person who probably did more than anyone to reveal this was Mircea Steriade, a Romanian-born neuroscientist who spent much of his career at Laval University in Quebec. Steriade was one of those researchers who combined extraordinary patience with deep technical skill. Starting in the 1980s and continuing through the 1990s and into the 2000s, he and his collaborators painstakingly recorded from individual neurons in the thalamus and cortex of animals, mapping out exactly how these cells behaved during different states: waking, drowsiness, light sleep, deep sleep. And what he found was remarkable. Here's how it works, and I want to walk you through this slowly, because it's genuinely one of the most elegant things in neuroscience. Thalamic neurons, the individual nerve cells in the thalamus, have a special property. They can fire in two completely different modes. In one mode, which neuroscientists call tonic firing, the neuron behaves like a reliable messenger. A signal comes in, the neuron fires in proportion to that signal, and it faithfully relays the information up to the cortex. This is what happens when you're awake and alert. Your thalamus is in relay mode, letting sensory information flow through cleanly. But here's where it gets interesting. When conditions change, specifically when the neuron's membrane potential drops below a certain threshold, which happens naturally as arousal decreases, these same thalamic neurons switch into a completely different mode called burst firing. Instead of firing steadily in response to incoming signals, they start producing rhythmic bursts of activity, little clusters of rapid-fire spikes, followed by a pause, then another burst, then another pause. And these bursts happen at very specific frequencies. The rhythm of those bursts, it turns out, is what generates sleep spindles and contributes to the slow oscillations of deep sleep. The mechanism behind this involves a specific type of ion channel called a T-type calcium channel. Now, I know that sounds technical, but the concept is actually pretty intuitive. Ion channels are tiny protein gates embedded in the membrane of every neuron. They open and close to let charged particles, ions, flow in and out of the cell, and that flow of charge is what creates electrical signals. Most ion channels open when the neuron is activated, when its voltage goes up. But T-type calcium channels are different. They're what's called low-threshold channels, and they have this peculiar property. They can only be activated after the neuron has been inhibited, after its voltage has dropped down for a while. It's like a spring-loaded mechanism. Push the voltage down, hold it there, and these channels get primed. Then, when the voltage starts to come back up even a little bit, the T-type channels snap open, calcium rushes into the cell, and you get a burst of firing. Then the channels close again. The voltage drops, and the whole cycle repeats. Think about what that means. You've got a neuron that, simply because of the types of ion channels it contains, naturally tends to oscillate when conditions are right. It's not that something external is forcing it to fire rhythmically. The rhythm is intrinsic to the cell itself. It emerges from the biophysics of the membrane. And when you have thousands and thousands of these thalamic neurons doing this together, connected to each other and to cortical neurons through feedback loops, you get these large-scale oscillations, the sleep spindles, the delta waves, that Loomis first recorded back in the 1930s without having any idea what was generating them. Steread called the thalamus the pacemaker of the sleeping brain, and that term really captures it. Just like the pacemaker cells in your heart generate rhythmic electrical impulses that coordinate heartbeats, thalamic neurons generate rhythmic bursts that coordinate cortical activity during sleep. And here's the really beautiful part. When the thalamus switches into burst mode, it's not just generating rhythms, it's also effectively disconnecting you from the outside world, because those bursts don't faithfully relay sensory information anymore. The signal gets chopped up, fragmented. So sounds in your room, light changes, minor physical sensations, they still reach your thalamus, but they don't make it through to your cortex in any coherent way. The thalamus has essentially closed the gate. That's why you can sleep through a steady hum of traffic, but might wake up to your name being called. The gating isn't absolute, and certain signals, especially ones your brain has learned to flag as important, can still punch through. Now, the thalamus doesn't do this alone. There's a thin shell of neurons surrounding the thalamus called the thalamic reticular nucleus, and this structure plays a crucial role. The reticular nucleus is made up almost entirely of inhibitory neurons, neurons that suppress the activity of other neurons, and it's wired in a very specific way. Cortical neurons send connections down to the thalamus, and on their way, they send branches to the reticular nucleus. The reticular nucleus, in turn, sends inhibitory connections back to the thalamic relay neurons. So you've got this feedback loop. Cortex talks to thalamus. Thalamus talks back to cortex, and the reticular nucleus sits in between, modulating the whole conversation. During wakefulness, the reticular nucleus is relatively quiet, and thalamic relay neurons are in tonic mode, faithfully passing information. But as you start to fall asleep, the reticular nucleus becomes more active, sending stronger inhibitory signals to the relay neurons. That inhibition pushes the relay neurons' voltage down, which primes those T-type calcium channels we talked about, which triggers burst firing, which generates sleep spindles. And those spindles, in turn, activate the reticular nucleus even more, creating a self-reinforcing cycle. It's a cascade. Once it starts, it tends to sustain itself, pulling you deeper into sleep. This is one of those things that, once you understand it, you start to appreciate just how precisely engineered, for lack of a better word, the brain's sleep system really is. It's not that sleep is simply the absence of wakefulness, like turning off a light switch. Sleep is an actively generated state, produced by specific circuits with specific biophysical properties, and those circuits have been shaped by hundreds of millions of years of evolution to do exactly what they do. But let's zoom out a bit, because the thalamus is only part of the story. The cortex itself, that wrinkled outer layer of the brain where most of your higher cognitive processing happens, also has intrinsic oscillatory properties. And understanding those required yet another line of research, one that focused on a different kind of neuron. You've probably heard that the brain has excitatory and inhibitory neurons. Excitatory neurons, when they fire, make their target neurons more likely to fire. Inhibitory neurons do the opposite. They suppress activity. And the interplay between these two types is absolutely fundamental to how brain rhythms work. Think of it like a seesaw, or maybe like breathing. Excitation pushes activity up, inhibition pulls it back down, and the alternation between the two creates a natural rhythm. The key inhibitory neurons in the cortex are called interneurons, and many of them use a neurotransmitter called GABA. These GABAergic interneurons come in different flavors. There are fast-spiking basket cells, chandelier cells, and others, and they connect to large populations of excitatory neurons. When a group of excitatory neurons fires, they activate nearby interneurons. Those interneurons then fire back, inhibiting the excitatory neurons. The excitatory neurons go quiet for a moment, which means the interneurons lose their input and go quiet too, which releases the excitatory neurons from inhibition, and they start firing again. And around it goes. This push and pull dynamic, excitation followed by inhibition followed by excitation, is one of the most fundamental mechanisms for generating oscillations in neural circuits, and the frequency of the oscillation depends on the specific properties of the neurons involved. How fast do the inhibitory neurons respond? How long does the inhibition last? How quickly do the excitatory neurons recover? Change any of those parameters and you change the frequency. Fast interneurons with brief inhibitory effects produce high-frequency oscillations, gamma waves, in the 30 to 100 Hz range, which we haven't talked about much yet, but which turn out to be incredibly important. Slower dynamics produce lower frequencies, theta, alpha, delta. And this is where a whole new chapter of the brainwave story opens up, because gamma oscillations, these fast, subtle rhythms that are much harder to detect on a scalp EEG than the big slow waves, started attracting serious attention in the late 1980s and 1990s. A German neuroscientist named Wolf Singer, working at the Max Planck Institute for Brain Research in Frankfurt, published a series of influential papers starting around 1989 that proposed something provocative. Singer and his colleague Charles Gray had been recording from the visual cortex of cats, and they noticed that when the cat was looking at a coherent visual object, say, a single moving bar of light, neurons in different parts of the visual cortex that were responding to different features of that object would synchronize their firing in the gamma range, around 40 Hz. Neurons responding to the orientation of the bar would fire in sync, with neurons responding to its motion, even though these neurons were in different cortical columns, sometimes millimeters apart. Singer proposed that this gamma synchronization was the brain's solution to what's called the binding problem, one of the deepest puzzles in neuroscience. The binding problem is this. When you look at, say, a red ball rolling across a table, different parts of your brain process the color, the shape, the motion, and the location separately. There's no single neuron or single brain area that puts it all together into one unified perception of red ball rolling. So how does the brain bind all those features into a single coherent experience? Singer's answer was temporal synchrony. The features are bound together because the neurons representing them fire at the same time, synchronized in the gamma band. Neurons that fire together wire together, as the saying goes, but more immediately, neurons that fire together are perceived together. This was a big deal. It suggested that gamma oscillations weren't just background noise or a byproduct of neural activity. They were a fundamental mechanism of perception, a way the brain organizes information, and it connected brain waves directly to the hard problem of consciousness. How does the brain create subjective experience? If gamma synchrony is how the brain binds features into unified percepts, then maybe the rhythms themselves are part of what it means to be conscious. Now, this idea has been debated intensely for decades. Not everyone was convinced by Singer's binding by synchrony hypothesis. Some researchers pointed out that gamma oscillations can be produced by simple mechanisms that have nothing to do with binding, that they might be a byproduct of the excitatory-inhibitory interplay we just discussed rather than a functional signal. Others argued that the evidence from cat visual cortex didn't necessarily generalize to more complex cognitive processes in humans. The debate got heated at times, as scientific debates do, with careers and reputations riding on whether gamma synchrony was the key to consciousness or just another neural epiphenomenon. But regardless of where you come down on the binding question, what became undeniable through the 1990s and 2000s was that brain oscillations at multiple frequencies are deeply intertwined with cognitive function. And one of the most fascinating discoveries in this area involved something called cross-frequency coupling. The idea that different brain rhythms don't operate independently, but actually interact with each other in structured ways. And the particular form of cross-frequency coupling that got researchers most excited was something called phase-amplitude coupling, where the phase of a slow oscillation, say a theta wave, modulates the amplitude of a faster oscillation, say a gamma burst. In other words, the gamma activity rides on top of the theta wave, getting stronger at certain phases of the theta cycle and weaker at others. This was first described in detail in the hippocampus, that memory structure we mentioned earlier. And the implications were really profound, because here's what that pattern suggested. The hippocampus might be using this nested rhythm, gamma packets tucked inside theta cycles, as a way to organize information in time. Think of it like this. Imagine the theta wave as a slow, rolling wave in the ocean, rising and falling maybe 5 or 6 times per second. Now imagine that on the crest of each of those waves, there's a little burst of fast ripples. Those are the gamma oscillations. Each gamma burst can carry a discrete chunk of information, a snapshot of neural activity representing something specific. And because the theta wave has a consistent rhythm, those gamma bursts arrive in a predictable sequence. So what you get is a temporal scaffolding, a way for the brain to line up multiple pieces of information, one after another, within a single theta cycle. This was a big deal. Researchers like Georgi Buzaki, working at Rutgers and later at NYU, became some of the most influential voices in explaining why this matters. Buzaki had been studying hippocampal oscillations since the 1980s, and by the mid-2000s he was articulating a vision of brain rhythms that went far beyond anything Berger or even Stereod had imagined. His argument, laid out beautifully in his 2006 book, Rhythms of the Brain, was that oscillations aren't just a side effect of neural activity. They're the brain's fundamental mechanism for organizing information, and phase-amplitude coupling was Exhibit A. Here's the specific claim, and it's worth slowing down to appreciate it. When you're navigating a space, say you're a rat running through a maze, because a lot of this work was done in rats, your hippocampus is generating a strong theta rhythm, around 6 to 8 hertz, and within each theta cycle, individual place cells fire at specific phases. A place cell, if you haven't encountered the term, is a neuron that fires when you're in a particular location. John O'Keefe discovered them in the early 1970s in London, and they're one of the most remarkable things in all of neuroscience. Individual neurons that act like GPS coordinates. O'Keefe would later share the Nobel Prize for this in 2014. But what researchers found in the 1990s, particularly John O'Keefe and Michael Recchi in a landmark 1993 paper, was that these place cells don't just fire when you're in their preferred location. They fire at progressively earlier phases of the theta cycle as you move through that location. This phenomenon is called theta phase precession, and it's one of the most elegant things the brain does. Let me try to make this concrete. Imagine you're walking down a hallway. As you approach a particular spot, the place cell for that spot starts firing, but it fires late in the theta cycle, near the trough, let's say. As you move into the spot, the cell fires earlier and earlier in each successive theta cycle, until by the time you're leaving that spot, it's firing near the peak. So the phase of the theta wave at which the cell fires encodes your position relative to that place field. It's not just where you are, it's where you are within a trajectory. The timing carries information that the firing rate alone doesn't. Now, combine this with phase-amplitude coupling. You've got multiple place cells, each representing a different location, each firing at a slightly different phase of the theta cycle. Within a single theta cycle, which lasts maybe 125 milliseconds, barely a blink, the hippocampus can represent a compressed sequence of locations. The place representing where you just were fires at one phase. The place representing where you are now fires at another. And the place representing where you're about to be fires at yet another. All within one sweep of the theta wave. It's like the brain is playing a tiny fast-forward movie of your trajectory, over and over, several times per second. And this, Buzzacki and others argued, is how the hippocampus supports memory. Not just spatial memory, but memory in general. Because if the hippocampus can compress sequences of events into the phase structure of a single oscillatory cycle, then it has a mechanism for binding together things that happened at different times into a coherent episode. That dinner you had last Tuesday, the taste of the food, the sound of your friend's laugh, the moment you knocked over your water glass. These events unfolded over minutes, but the hippocampus can replay them in compressed form, nested within its oscillatory rhythms, and that compressed replay might be exactly what consolidates the memory. And here's where it connects back to sleep, in a way that would have astonished those early researchers at Tuxedo Park. Remember the slow oscillations of deep sleep, those big rolling delta waves? And remember the sleep spindles, those bursts generated by the thalamic reticular nucleus? Well, it turns out that during slow wave sleep, the hippocampus produces its own characteristic pattern called sharp wave ripples. These are brief, intense bursts of activity. The ripple component oscillates at around 150 to 200 hertz, which is faster than anything we've talked about so far. And during these ripple events, place cells replay sequences of locations that the animal experienced while it was awake. the same cells fire in the same order, but compressed into a tiny fraction of the original time. A journey that took seconds gets replayed in tens of milliseconds. Now here's the thing that really ties it all together. These hippocampal ripples don't happen randomly during sleep. They tend to occur at specific phases of the slow oscillation. During the up state, when cortical neurons are depolarized and ready to be activated. And the sleep spindles, those thalamic bursts, also tend to nest within the up state of the slow oscillation. So you get this remarkable three-level nesting. The slow oscillation provides the broadest temporal frame. The sleep spindle nests within it, and the hippocampal ripple nests within the spindle. It's cross-frequency coupling across three different frequency bands, coordinated across three different brain structures: cortex, thalamus, and hippocampus, all working together in a precisely timed sequence. And the function? Memory consolidation. The current understanding, supported by work from many labs, but particularly championed by researchers like Jan Born at the University of Tübingen, is that this nested oscillatory sequence is how the brain transfers memories from the hippocampus to the cortex for long-term storage. The hippocampus replays the memory during the ripple. The spindle provides a window of enhanced cortical plasticity, a moment when cortical synapses are primed to be strengthened. And the slow oscillation coordinates the whole thing, ensuring that the hippocampal replay arrives at the cortex at exactly the right moment. Born and his colleagues showed in a series of elegant experiments in the 2000s and 2010s that if you enhance slow oscillations during sleep, using gentle electrical stimulation or auditory tones timed to the rhythm, you can actually improve memory consolidation. People remember more of what they learned the day before. The oscillations aren't just correlated with memory, they're causally involved. Think about what that means for a second. Those slow, drowsy brainwaves that Loomis recorded back in 1937, the ones he just categorized as stages of sleep. They're not passive. They're not the brain idling. They're the brain doing some of its most critical work. Every night, while you're lying there unaware, your brain is methodically replaying the day's experiences, transferring them from temporary hippocampal storage to more permanent cortical networks, and it's using the precise timing relationships between oscillations at different frequencies to do it. The rhythm is the mechanism. This realization that oscillations aren't just markers of brain states, but active computational tools, really crystallized in the 2000s and transformed how neuroscientists think about brain waves. It moved the field from, what frequency do we see in this condition, to, what is this rhythm actually doing, mechanistically, to neural computation. And one of the key conceptual frameworks that emerged was the idea of communication through coherence, proposed by Pascal Frias, a neuroscientist working at the Ernst Strungmann Institute in Frankfurt. Fries published an influential paper in 2005, and then a more developed version in 2015, arguing that oscillatory synchrony between brain regions is the mechanism by which those regions communicate effectively. The idea goes like this. Two brain regions might be anatomically connected. There might be axons running from one to the other, but that doesn't mean they're always communicating effectively. For signals from region A to influence region B, the neurons in region B need to be in a receptive state when those signals arrive. And what determines whether they're in a receptive state? Their oscillatory phase. If region B's neurons are at the excitable phase of their oscillatory cycle when input from region A arrives, the signal gets through. If they're at the inhibitory phase, the signal gets suppressed. So for effective communication, the oscillations in regions A and B need to be coherent. They need to be synchronized so that the excitable windows line up. This is elegant because it means the brain can dynamically route information just by changing which regions are oscillating in sync. No need to grow new connections or change the wiring. Just shift the phase relationship, and suddenly, region A is talking to region C instead of region B. Fries proposed that different frequency bands might serve different communication functions. Gamma synchrony for feed-forward communication, carrying sensory information up the cortical hierarchy, and beta synchrony for feedback communication, carrying top-down predictions and expectations back down. It's a beautiful framework, and while it's still being tested and refined, it gave the field a concrete, mechanistic hypothesis for why the brain oscillates at all. And this brings us to something that had been lurking in the background of brainwave research for decades, a question that's both practical and deeply important. If oscillations are this central to how the brain works, what happens when they go wrong? Because they do go wrong, and the consequences can be devastating. One of the earliest and most dramatic examples is epilepsy, which has been intertwined with the story of brainwaves from the very beginning. Remember, Berger's original EEG recordings showed abnormal patterns in patients with brain lesions, and by the late 1930s, neurologists were already using EEG to identify epileptic foci, regions of the brain where seizures originate. But understanding what a seizure actually is in terms of oscillatory dynamics took much longer, and the story involves some of the most fundamental questions about how neurons coordinate, and how that coordination can catastrophically break down. So let's talk about what a seizure actually looks like at the level of brain waves, because it's not just noise, it's not static. In many ways, a seizure is the opposite of chaos. It's too much order. Think about that for a second. We've spent all this time talking about how the brain uses oscillations to organize information, how different frequencies carry different signals, how phase relationships route communication between regions. A healthy brain is this incredibly complex symphony of rhythms, all interleaving, all doing different things at different times. A seizure is what happens when that symphony collapses into everyone playing the same note at the same time. It's hypersynchrony, massive populations of neurons locking into the same rhythm, firing together in a way that overwhelms normal function. On an EEG, you can actually watch it happen. In a generalized tonic-clonic seizure, what people used to call a grand mal seizure, the recording goes from normal background activity to these enormous rhythmic spikes and waves that dominate every channel. The amplitude shoots up because so many neurons are firing in lockstep. And here's what's really striking. The frequency often starts high, maybe in the beta range or even faster, and then progressively slows down as the seizure evolves. You get these characteristic 3-per-second spike and wave complexes in absent seizures, the kind where a person just blanks out for a few seconds, stares into space, and then comes back. Frederick Gibbs and Erna Gibbs, working with William Lennox at Harvard in the mid-1930s, were among the first to document that specific 3 Hz pattern and connect it to absence epilepsy. That was a huge deal. For the first time, you had a brainwave signature that could reliably distinguish one type of seizure from another, and it turned EEG from a curiosity into a genuine clinical tool. But understanding why neurons hypersynchronize, that took decades of work, and it gets right to the heart of excitatory-inhibitory balance, which we touched on earlier. Remember how we talked about the interplay between excitatory pyramidal neurons and inhibitory interneurons, how their back and forth creates oscillations. In a healthy brain, the inhibition acts as a brake. Excitatory neurons fire, inhibitory neurons respond and tamp things down, and you get this controlled rhythm. In epilepsy, that brake fails. Maybe the inhibitory interneurons aren't working properly. Maybe they've been damaged, or the GABA receptors they signal through aren't functioning right. Maybe the excitatory connections have been strengthened through some pathological process. Either way, excitation wins, and when one group of neurons fires, instead of being pulled back by inhibition, the excitation spreads to neighboring neurons, which fire too, which excite their neighbors, and you get this runaway cascade. It's like a crowd doing the wave in a stadium, except instead of a fun ripple that passes through, every single person stands up and never sits back down. And here's where it connects back to the thalamus in a really important way. Remember Mircea Stariade's work on thalamic oscillations, the T-type calcium channels, the burst firing mode, the thalamic reticular nucleus generating sleep spindles? It turns out the thalamocortical circuits that produce those beautiful, organized sleep rhythms are also deeply involved in certain types of seizures. Absence seizures in particular seem to hijack the same thalamocortical loop that generates spindles. The thalamic reticular nucleus, which normally helps gate sensory information and produce those 12 to 14 Hz spindle bursts during sleep, gets caught in a pathological version of that rhythm. The T-type calcium channels that enable burst firing become part of the problem. They help sustain the abnormal oscillation. This is why some anti-epileptic drugs for absence seizures, like ethosuximide, work specifically by blocking T-type calcium channels. You're targeting the very mechanism that generates the pathological rhythm. It's one of the clearest examples in all of neuroscience where understanding the oscillatory mechanism directly led to a treatment. Now, focal seizures, seizures that start in one specific region of the brain, tell a different but equally fascinating story. These are the ones where identifying the seizure focus became a matter of life-changing importance. Because if you can find where the seizure starts, a neurosurgeon can sometimes remove that tissue and cure the epilepsy entirely. This is where EEG became not just diagnostic, but surgical. Wilder Penfield, working at the Montreal Neurological Institute starting in the 1930s and continuing for decades, pioneered this approach. He would open the skull of awake patients. The brain itself has no pain receptors, so this is possible with local anesthesia to the scalp, and use electrodes placed directly on the cortical surface to map out which areas produced seizure activity and which areas controlled critical functions like speech and movement. He'd electrically stimulate different spots and ask the patient what they experienced. Sometimes they'd report a tingling in their hand, or they'd suddenly recall a vivid memory, or they'd hear music. Penfield was essentially creating a functional map of the living human brain, one electrode at a time, and using that map to guide his surgical cuts. This technique, electrocorticography, or ECOG, gave researchers access to brain waves with much higher fidelity than scalp EEG. You're recording right from the surface of the brain, no skull in the way, no scalp muscles adding noise. The signals are cleaner, sharper, and you can detect high-frequency activity that scalp EEG simply can't resolve. And what they found was remarkable. In the tissue surrounding an epileptic focus, neurons showed what are called interictal spikes, brief, sharp electrical discharges that happen between seizures. These spikes became the signature that neurosurgeons looked for when deciding what tissue to remove. But here's the thing. Not every region that shows interictal spikes is actually part of the seizure-generating zone. Some of those spikes are just echoes, reflections of abnormal activity propagating from elsewhere. Distinguishing the true epileptogenic zone from irritated bystander tissue remains one of the hardest problems in epilepsy surgery to this day. And this challenge pushed the technology forward in ways nobody expected. By the 1990s and 2000s, researchers were developing dense electrode grids, arrays of 64, 128, even 256 electrodes placed directly on the brain surface to get ever more precise maps of seizure propagation. They could watch a seizure unfold in slow motion, see exactly how the hypersynchronous wave traveled from its origin point across the cortex, identify the pathways it used, the barriers it encountered. Some seizures spiral outward like a ripple in a pond. Others follow specific anatomical pathways, jumping along white matter tracks to distant regions. The dynamics are complex and beautiful in a terrible way. Beautiful because the physics of wave propagation is elegant. Terrible because each of those waves represents a person losing consciousness, losing control of their body, sometimes losing minutes of their life that they'll never remember. But let's step back from epilepsy for a moment, because the story of oscillations going wrong extends far beyond seizures. And this is where things get really interesting and really complicated, because by the late 20th century, researchers started finding abnormal oscillatory patterns in almost every major neurological and psychiatric condition you can name. Parkinson's disease, and this one is particularly striking. In the late 1990s and early 2000s, Peter Brown, working first at the Institute of Neurology in London and later at Oxford, made a discovery that reshaped how people thought about the disease. Parkinson's, as you probably know, involves the loss of dopamine-producing neurons in a deep brain structure called the substantia nigra. The classic symptoms are tremor, rigidity, and slowness of movement. For decades, the explanation was purely chemical. You lose dopamine, the motor circuits don't work properly. End of story. But Brown and his colleagues had access to something new: recordings from deep brain stimulation electrodes that had been implanted in Parkinson's patients. Deep brain stimulation, or DBS, was already being used as a treatment. Surgeons would implant electrodes in the subthalamic nucleus or the globus pallidus, deliver high-frequency electrical stimulation, and somehow, remarkably, the symptoms would improve. But nobody fully understood why it worked. What Brown found was that the basal ganglia of Parkinson's patients were dominated by exaggerated beta oscillations, activity in the 13 to 30 Hz range that was far stronger and more persistent than in healthy brains. And here's the kicker. The severity of the beta activity correlated with the severity of the motor symptoms. More beta, more rigidity, more slowness. When patients took their dopamine medication and their symptoms improved, the beta power dropped. When DBS was turned on and symptoms improved, beta power dropped. It was as if the motor circuits were stuck in a beta state, locked into a rhythm that was supposed to be there some of the time, during postural holding and motor suppression. But that had become pathologically dominant, drowning out the faster gamma activity needed for initiating and executing movements. Think about what that means in terms of everything we've talked about. Remember Fries' communication through coherence framework: gamma for feed forward, beta for feedback. In Parkinson's, it's as if the feedback channel is permanently jammed open. The motor system is perpetually in a state of top-down suppression, sending hold still, don't move signals that the patient can't override. The oscillatory framework suddenly wasn't just an abstract theory about information routing. It was explaining the lived experience of a devastating disease. And DBS, that mysterious treatment that nobody quite understood, might work precisely because the high-frequency stimulation disrupts the pathological beta synchrony, breaks the lock, and allows the motor circuits to shift back into the dynamic, flexible oscillatory patterns they need. Brown published key papers on this in the early 2000s, and the idea took off. Suddenly, people were looking at oscillatory signatures in every movement disorder, every psychiatric condition. And what they found was both exciting and humbling. In schizophrenia, researchers had been noticing abnormal gamma oscillations since the late 1990s. The connection to the binding by synchrony hypothesis was immediate and tantalizing. If gamma synchrony is how the brain binds features into coherent perceptions, and if gamma is disrupted in schizophrenia, could that explain the fragmented perception, the disordered thinking, the hallucinations? The evidence pointed toward dysfunction in the fast-spiking parvalbumin-positive interneurons, the same inhibitory cells that are critical for generating gamma rhythms. These cells seem to be compromised in schizophrenia, possibly due to problems with NMDA receptor signaling. And here's a detail that ties it together beautifully. Ketamine, a drug that blocks NMDA receptors, produces symptoms that look remarkably like schizophrenia in healthy volunteers, and it does so while disrupting gamma oscillations. The pharmacology, the physiology, and the clinical symptoms all converge on the same oscillatory mechanism. But I want to be careful here, because this is exactly the point where the story gets seductive and potentially misleading. Finding abnormal oscillations in a disease doesn't automatically mean the oscillations are causing the disease. They might be a consequence, a downstream effect of something else going wrong, or they might be compensatory, the brain's attempt to work around a problem. Untangling cause from correlation in oscillatory disorders is one of the great ongoing challenges, and it requires exactly the kind of causal manipulation tools that are only now becoming available. Optogenetics, for instance, the technique developed in the mid-2000s by Carl Dieserath at Stanford and Ed Boyden at MIT, where you genetically modify specific neurons to respond to light so you can turn them on or off with millisecond precision. For the first time, you could ask, if I artificially impose a specific oscillatory pattern on a brain circuit, does it produce a specific behavioral effect? And the answer, in study after study, has been yes. Induce theta synchrony between the hippocampus and prefrontal cortex, and memory performance improves. Disrupt gamma in the visual cortex, and perceptual binding falls apart. Impose pathological beta in the motor circuits, and movement slows down. These causal experiments have been transformative, but they've also revealed just how much we still don't understand. Because the brain doesn't have one oscillation doing one thing. It has dozens of oscillatory patterns interacting across multiple spatial and temporal scales simultaneously, and perturbing any one of them ripples through the whole system in ways that are hard to predict. Which brings us to something that's been quietly revolutionizing the field. Not a new discovery about the brain itself, but a new way of analyzing the signals we've been recording all along. And it starts with a mathematician's insight about how signals are built from simpler pieces. The mathematician's name was Joseph Fourier, and he lived in France at the turn of the 19th century, born in 1768, orphaned young, educated by Benedictine monks, eventually swept up in the French Revolution, nearly guillotined, and then somehow ended up accompanying Napoleon on the Egyptian campaign. Fourier's life alone could fill an evening, but the part that matters for us is a paper he presented to the French Academy of Sciences in 1807, in which he made a claim so bold that some of the greatest mathematicians of his era, Lagrange among them, refused to believe it. Fourier said that any periodic signal, no matter how complicated, no matter how jagged or smooth or strange its shape, could be broken down into a sum of simple sine waves. Pure, clean, repeating sine waves of different frequencies, different amplitudes, and different phases, all added together. Layer enough of them on top of each other, and you could reconstruct any signal you wanted. Think about what that means for brainwaves. When you look at a raw EEG trace, that squiggly line that Berger first recorded back in the 1920s, it looks like chaos. It looks like someone scribbling with a pen during an earthquake. But Fourier's insight tells you that buried inside that messy signal are individual frequency components. There's some alpha in there, some theta, maybe a burst of gamma, some slow delta underneath it all. They're all superimposed, all riding on top of each other, and the raw trace is just their sum. If you could somehow pull them apart, separate them back into their individual frequency components, you'd see the building blocks. You'd see what the brain is actually doing. The mathematical tool for doing this, the Fourier transform, has been around in various forms since Fourier's original work, but it wasn't practical for analyzing brain signals until computers came along. In the 1960s, a pair of mathematicians named James Cooley and John Tukey published an algorithm called the Fast Fourier Transform, or FFT, in 1965. And this changed everything, not just for neuroscience, but for signal processing across every field you can think of: telecommunications, audio engineering, radar, medical imaging. The FFT made it computationally cheap to take a chunk of signal and decompose it into its frequency components. Suddenly, instead of staring at a raw EEG trace and trying to eyeball whether there was more alpha or more theta, you could run the FFT and get a precise power spectrum, a graph showing exactly how much energy was present at each frequency. You could quantify it. You could compare it across conditions, across patients, across brain regions. And this was genuinely useful. Sleep researchers could now precisely measure how much slow-wave activity someone had in a given sleep stage. Clinicians could look at the power spectrum of a patient's EEG and see if there was an abnormal peak at 3 Hz. That signature of absence seizures we talked about. Researchers studying attention could track changes in alpha power when someone closed their eyes versus opened them. The FFT gave the field numbers where before there had been subjective impressions. But here's the thing about the Fourier transform, and this is subtle, so let's take it slow. The classic FFT assumes that your signal is stationary. That means it assumes the frequency content doesn't change over time. You feed it a chunk of EEG, say 10 seconds, and it gives you back the average frequency content across that entire 10-second window, which is fine if the signal really is stable over that period. But brain signals aren't stable. They change moment to moment. A burst of gamma might last only 200 milliseconds. A sleep spindle comes and goes in about a second. If you average over 10 seconds, that spindle gets smeared out. Its energy is still there in the power spectrum, but you've lost the information about when it happened. And in the brain, when something happens is often just as important as whether it happens at all. So people started looking for ways to get both frequency information and time information simultaneously. One approach is straightforward. You just chop the signal into short windows and run the FFT on each one separately. This is called the short-time Fourier transform, and it works, but it forces you into a trade-off. If you make the windows short, you get good time resolution. You can see when things happen. But your frequency resolution gets blurry. If you make the windows long, your frequency resolution sharpens up, but you lose the ability to pinpoint events in time. It's like a fundamental uncertainty principle for signal analysis. You can't have perfect resolution in both dimensions at once. And this is where another mathematical idea enters the picture, one that's become enormously important in modern neuroscience: wavelet analysis. The core idea behind wavelets is elegant. Instead of decomposing your signal into infinite sine waves like Fourier does, you decompose it into little localized wave packets. Each wavelet is a brief oscillation that starts, peaks, and dies away. And the clever trick is that you can stretch or compress these wavelets to match different frequencies. A stretched-out wavelet captures low-frequency content, and because it's long, it gives you good frequency resolution at low frequencies. A compressed wavelet captures high-frequency content, and because it's short, it gives you good time resolution at high frequencies. The trade-off between time and frequency resolution is still there. You can't escape it, but wavelets distribute that trade-off in a way that's actually well matched to how brain oscillations behave. Low-frequency rhythms like delta and theta tend to be sustained, so you don't need pinpoint time resolution for them. High-frequency rhythms like gamma tend to come in brief bursts, so you need good time resolution, but can tolerate some frequency smearing. Wavelets give you exactly that. By the 1990s and 2000s, wavelet analysis and related time-frequency methods had become standard tools in the neuroscience toolkit. Researchers could now create beautiful time-frequency maps of brain activity, spectrograms essentially, showing how the power at each frequency evolved over time. And when you look at one of these spectrograms during, say, a transition from waking to sleep, it's remarkable. You can watch the alpha rhythm fade, see theta activity emerge, watch the first sleep spindles appear as brief bursts of sigma band energy, and then see the deep, slow waves of delta rise up like a tide. It's the architecture of sleep, made visible in a completely new way. But even time-frequency analysis has limitations, and this is where the story gets really interesting. Because all of these methods, Fourier, wavelets, spectrograms, they all assume something fundamental. They assume that the signal you're analyzing is generated by a linear system, that the different frequency components are independent, that they just add together like Fourier said. And in many physical systems, that's a reasonable assumption. But the brain is not a linear system, not even close. The brain is wildly, profoundly nonlinear. Neurons have thresholds. They don't respond at all until their input reaches a certain level, and then they fire. Synaptic transmission involves cascades of biochemical reactions with saturation points and feedback loops. The interactions between brain regions are recurrent and self-modifying, and all of this nonlinearity means that the different frequency components in the EEG are not independent. They interact, they modulate each other, they couple. We touched on this earlier, remember, cross-frequency coupling, the way gamma oscillations nest inside the troughs of theta waves in the hippocampus. That's a nonlinear interaction. The amplitude of the fast rhythm is being modulated by the phase of the slow rhythm. A standard Fourier analysis won't show you that. It'll show you that there's theta power and there's gamma power, but it won't tell you that they're linked, that the gamma bursts are locked to a specific phase of the theta cycle. To see that, you need specialized analyses, phase amplitude coupling measures, bicoherence, mutual information, tools that were developed specifically to capture these nonlinear relationships. And this brings us to something that's been a quiet revolution in the field over the last couple of decades: the realization that it's not just the power in different frequency bands that matters. It's the relationships between them, the coupling, the coherence, the timing. Two brain regions might both show strong gamma activity, but the critical question is whether their gamma oscillations are synchronized with each other. Are they oscillating in phase? Are they coherent? Because that's what determines whether they're actually communicating, at least according to the communication through coherence framework we explored earlier. And measuring that coherence requires going beyond simple power spectra into the realm of connectivity analysis. So now, imagine you've got not just one or two EEG electrodes, but 64, or 128, or 256, spread across the entire scalp. Modern high-density EEG systems routinely use these numbers. And for each pair of electrodes, you can compute coherence at every frequency band. You can ask, is the gamma activity over the left frontal cortex synchronized with the gamma activity over the right parietal cortex? Is the theta rhythm in the temporal regions coupled to the alpha rhythm in the occipital regions? You start building a picture not just of what each brain region is doing in isolation, but of how they're talking to each other. And this is what people mean when they talk about the functional connectome, the map of dynamic communication patterns across the brain. The mathematics here gets sophisticated, and honestly, a lot of it draws on tools from graph theory and network science that were originally developed for completely different purposes, analyzing social networks or internet traffic or power grids. The brain, it turns out, has a network architecture that shares some deep structural properties with these other complex systems. It has hubs, highly connected regions that serve as relay points. It has modules, clusters of regions that are densely connected to each other, but more loosely connected to other clusters. And it has what's called small world topology, meaning that even though most connections are local, there are enough long-range connections that information can get from any region to any other region in surprisingly few steps. This was described in a landmark paper by Danielle Bassett and Ed Bullmore, among others, and it's become one of the organizing frameworks for understanding how the brain balances the competing demands of specialized local processing and integrated global communication. Here's why this matters for brainwaves specifically. Those oscillatory patterns we've been talking about all night, the alpha, theta, gamma, delta, all of it, they're not just local phenomena. They're the mechanism by which the network operates. The oscillations are the communication protocol. When two modules of the brain need to exchange information, they synchronize their oscillations. When they need to operate independently, they desynchronize. And the flexible, moment-to-moment reconfiguration of these synchronization patterns is what allows the brain to adapt to whatever task it's facing, whether that's focusing on a conversation, navigating a crowded street, solving a math problem, or letting go of the day and drifting into sleep. And speaking of sleep, let's bring this back to where you are right now, lying in the dark, letting your mind wander. Because some of the most fascinating applications of these modern analytical tools have been in understanding what the sleeping brain is actually doing with its oscillations. Remember those sharp wave ripples we talked about, the ones that replay compressed memories during deep sleep? When researchers applied modern connectivity analyses to sleeping brains, they discovered something remarkable. During those ripple events, the communication patterns across the brain briefly reconfigure in a very specific way. The hippocampus, which is normally somewhat isolated during deep sleep, suddenly becomes a broadcasting hub. For a fraction of a second, it sends coordinated signals to widespread cortical regions, and the regions that receive the strongest signals are the same ones that were active during the original learning experience. It's as if the hippocampus is saying, Here, this is what happened today, and it's targeting the message to exactly the right recipients. And the timing is exquisite. The sharp wave ripples in the hippocampus tend to occur during a specific phase of the cortical slow oscillation, during the up state, when cortical neurons are depolarized and ready to receive input, and they often coincide with sleep spindles, those thalamocortical bursts we talked about earlier. So you get this nested temporal structure. The slow oscillation provides a broad timing framework. The spindle opens a window of cortical plasticity, and the ripple delivers the specific memory content within that window. 3 different oscillatory patterns, generated by 3 different brain structures, coordinating across multiple timescales to accomplish something none of them could do alone. This hierarchical coordination has been studied extensively by a number of groups, but some of the most compelling work has come from Jury Born's lab in Germany and Georgi Buzaki's group, who've shown that the precise temporal coupling between slow oscillations, spindles, and ripples predicts how well memories are consolidated overnight. People who show tighter coupling remember more the next morning. And as people age, this coupling tends to loosen. The oscillations still occur, but they're less precisely coordinated, and that may be part of why older adults often have more difficulty with memory consolidation during sleep. Which raises an obvious question, one that a lot of researchers and clinicians are now actively pursuing. If the oscillatory coordination is what matters, can we enhance it? Can we nudge the brain's rhythms into better alignment and actually improve the quality of what sleep does for memory? And this is where things get really exciting, and honestly a little bit science fiction feeling, even though it's all very real and happening right now in labs around the world. The idea is deceptively simple. If the slow oscillation is the master clock that organizes spindles and ripples, then what if you could listen to the brain while someone sleeps, detect the rhythm of those slow oscillations, and then deliver a gentle sensory stimulus, a quiet sound, say, timed precisely to the up state of the oscillation. Not to wake the person up, not even to be consciously heard, just a faint click or tone, arriving at exactly the right moment to reinforce the oscillation's natural rhythm, to give it a little nudge, like pushing a child on a swing at just the right point in the arc. This is called auditory closed-loop stimulation, and the person who really brought it into the spotlight is John Born, the same researcher we've been talking about, working with his colleague Hong Viet Ngo. Around 2013, Ngo and Born published a study that was beautifully elegant in its design. They had participants learn word pairs before going to sleep, a classic declarative memory task. Then, while the participants slept, the system monitored their EEG in real time, detected the slow oscillations, and played brief pink noise pulses timed to arrive during the upstate. The control condition used the same sounds but delivered them out of phase, or not at all. And what they found was striking. The in-phase stimulation didn't just boost the amplitude of the slow oscillations. It also increased spindle activity, as if reinforcing the slow oscillation caused the thalamus to respond with more robust spindle bursts. And the next morning, the people who received the properly timed sounds remembered significantly more word pairs than those who didn't. The oscillatory hierarchy we were just talking about, slow oscillation, spindle, ripple, was being externally enhanced, and it actually improved memory consolidation. Now, think about what that means for a second. You're not injecting information. You're not stimulating neurons directly. You're just playing a quiet sound at the right time, and the brain does the rest. It's like you're conducting an orchestra, except you're not telling the musicians what notes to play. You're just giving them a clearer downbeat so they can synchronize better on their own. The brain already knows what to do during sleep. it's already trying to consolidate memories. You're just helping it do it more effectively by tightening the temporal coordination that naturally tends to be a little loose and variable. And this approach has been replicated and extended by a number of other groups. Bernhard Starosina and colleagues have shown similar effects, and there's been work exploring whether the same principle can help older adults, the very population where that oscillatory coupling tends to degrade. The results there are still being refined, but early findings are genuinely promising. If aging loosens the coordination between slow oscillations and spindles, and if that loosening contributes to memory decline, then a non-invasive intervention that tightens it back up could be meaningful. Not a drug, not surgery, just sound, delivered with precise timing. But here's the thing. The timing really matters. And that's what makes closed-loop stimulation so different from the kind of brain stimulation you might have heard about in other contexts. Open-loop approaches, where you just deliver stimulation on a fixed schedule regardless of what the brain is doing, tend to be much less effective, and sometimes they even make things worse. If your sound pulse arrives during the down state of the slow oscillation instead of the up state, you can actually suppress the oscillation rather than enhance it. The brain's rhythms aren't just background noise that you can override. They're functional states, and the effect of any input depends critically on when it arrives relative to the ongoing oscillatory phase. This is that same principle of timing-dependent processing that keeps showing up throughout our whole story, from communication through coherence to phase-amplitude coupling. The brain is a system that cares deeply about when things happen, not just whether they happen. And auditory stimulation isn't the only game in town. There's also transcranial electrical stimulation, applying weak electrical currents to the scalp to try to entrain brain oscillations directly. Lisa Marshall, also working with Bourne's group, published some of the earliest work on this back in 2006, using slow oscillating transcranial direct current stimulation during sleep. The idea was similar. Drive the slow oscillation externally and see if memory consolidation improves. And it did, at least under certain conditions. But this approach has been harder to replicate consistently, partly because the electrical fields reaching the brain through the skull are extremely weak and diffuse, and partly because the relationship between the applied current and the resulting neural activity is complicated and not fully understood. It's a reminder that even when you have the right conceptual framework, oscillatory coordination matters, timing matters. The practical engineering of actually intervening is enormously challenging. So let's zoom out for a moment, because what's happening here represents a broader shift in how neuroscience thinks about brain oscillations, and it connects to something we should talk about. For most of the history we've been tracing, from Berger's first recordings, through the sleep staging work, through the discovery of gamma binding and theta-gamma coupling, oscillations were primarily something you observed. You recorded them, you analyzed them, you correlated them with behavior or cognition or disease. They were a window into the brain, a readout. But increasingly, the field has moved toward treating oscillations not just as indicators, but as mechanisms that can be targeted. If gamma synchrony is disrupted in schizophrenia, can you restore it? If beta oscillations are exaggerated in Parkinson's, can you suppress them? If slow oscillation spindle coupling is degraded in aging, can you tighten it? The shift is from oscillations as biomarkers to oscillations as therapeutic targets. And this shift has been accelerated enormously by a technology we touched on earlier: brain-computer interfaces, or BCIs. The basic concept has been around since the 1970s, when Jacques Vidal at UCLA first coined the term and demonstrated that EEG signals could be used to control a computer cursor. But for decades, BCIs were mostly laboratory curiosities, impressive demonstrations that didn't quite translate into practical tools. What changed was a combination of better recording hardware, better signal processing algorithms, and frankly, better machine learning. Modern BCIs can decode remarkably specific information from neural oscillations in real time. The most dramatic examples involve invasive recordings, electrode arrays implanted directly on or in the brain. The BrainGate project, which grew out of work by John Donahue at Brown University starting in the early 2000s, showed that people with paralysis could use signals from motor cortex to control robotic arms, move cursors, even type messages. And what are those signals? They're oscillatory patterns, beta desynchronization when you intend to move, gamma bursts encoding specific movement parameters, local field potentials reflecting the coordinated activity of neural populations. Everything we've talked about regarding how oscillations encode information, that's what BCIs are reading out and translating into commands. But there's a non-invasive side too, and it's more directly relevant to everyday life. EEG-based neurofeedback has been around since the 1960s, when Joe Kamiya at the University of Chicago showed that people could learn to voluntarily increase their alpha rhythm when given real-time feedback about their brain activity. The idea was tantalizing. If you could train people to control their own oscillations, maybe you could treat anxiety, ADHD, epilepsy, insomnia. And there's been a long and honestly somewhat contentious history around neurofeedback. Some studies have shown real benefits, particularly for attention and epilepsy. But the field has also been plagued by inadequate controls, small sample sizes, and a tendency toward overclaiming. It's one of those areas where the concept is sound, oscillations are functional, and learning to modulate them should in principle have cognitive or clinical effects. But the practical execution has been uneven. What's different now, and what's genuinely exciting, is that the tools have gotten so much better. Real-time EEG processing that would have required a room full of equipment in the 1990s can now run on a laptop or even a smartphone connected to a lightweight headband. Companies like Dream, a French startup that developed a headband specifically designed to deliver closed-loop auditory stimulation during deep sleep, have tried to bring the kind of research Born and Go pioneered directly to consumers. Whether these consumer devices deliver the same precision as laboratory systems is an open and important question, but the trajectory is clear. The science of brain oscillations is moving out of the lab and into bedrooms, clinics, and eventually, maybe into everyday wearable technology. And this brings us to something that I think is one of the most profound implications of everything we've been talking about. When you understand that brain oscillations aren't just epiphenomena, they're not just the hum of the engine, but are actually the mechanism by which the brain coordinates information processing, gates sensory input, consolidates memory, enables flexible communication between regions, then you start to see oscillations as something almost like a vital sign, not just heart rate, not just blood pressure, but oscillatory health. The quality of your sleep spindles, the precision of your theta-gamma coupling, the strength of your slow oscillations. These things reflect the functional integrity of your brain's communication infrastructure, and they change with age, with disease, with sleep deprivation, with stress. There's been fascinating work, for instance, showing that changes in sleep oscillations can be detected years before the clinical onset of neurodegenerative diseases. Disrupted sleep spindles and slow oscillations have been observed in people who are in the very early pre-symptomatic stages of Alzheimer's disease. Matthew Walker at UC Berkeley has been particularly vocal about the bidirectional relationship here. Not only does Alzheimer's pathology disrupt sleep oscillations, but the disrupted sleep may itself accelerate the accumulation of amyloid beta protein, creating a vicious cycle. The oscillations aren't just a symptom, they may be part of the causal chain. And if that's true, then monitoring and maintaining healthy sleep oscillations isn't just about feeling rested. It could be about long-term brain health in a really fundamental way, which connects to something that might seem mundane, but is actually deeply important: the simple act of getting good sleep. Everything we've talked about tonight, all these elegant mechanisms, the slow oscillations orchestrating spindles and ripples, the hippocampus replaying memories to the cortex, the thalamus gating sensory input so you can stay asleep. All of it requires time. It requires uninterrupted cycles of non-REM and REM sleep repeated across the night. When sleep is fragmented or shortened, these processes don't just get compressed, they get disrupted. The oscillatory coordination falls apart. The memory consolidation is incomplete. The sensory gating is impaired. And here's something that connects all the way back to where we started, to Hans Berger sitting in his lab in Jena, carefully pasting electrodes to his patients' scalps and recording those first wobbly traces of alpha rhythm. Berger believed, and he was right, even though he couldn't prove it with the tools he had, that the electrical activity of the brain was not random noise. It was organized, it was meaningful, and it reflected the inner workings of the mind. He spent years being dismissed, years being told that what he was recording was artifact, muscle twitch, equipment malfunction. And yet he kept recording, kept measuring, kept believing that those rhythmic waves meant something. Nearly a century later, we're still discovering just how right he was and how deep the meaning goes. So as you're lying here, in whatever comfortable position you've settled into, your own brain is doing something remarkable. It's beginning to shift its oscillatory patterns. If you're starting to feel drowsy, your alpha rhythm is probably fading, replaced by the slower, more irregular theta activity that marks the transition into sleep. And soon, if you let yourself drift, those sleep spindles will begin. Brief, beautiful bursts of 12 to 14 Hz activity, generated by the thalamic reticular nucleus, sweeping across your cortex. And beneath them, the slow oscillations will start their grand rolling rhythm, coordinating everything beneath them into a single, unified pulse of activity. The upstate bringing millions of neurons online together, the downstate letting them rest, and then the upstate again. Like the slowest, deepest breathing your brain knows how to do. And right now, in this moment, you don't have to do anything about any of it. That's the beautiful thing. Your brain knows how to do this. It's been doing it every night of your life since before you were born, actually. Fetuses show organized sleep-like oscillatory patterns in the womb. This isn't something you need to manage or optimize or worry about. The thalamus will do its thing. The cortex will do its thing. The hippocampus will sort through whatever happened today and decide what's worth keeping, replaying those experiences during sharp wave ripples that last maybe 1/10 of a second each, compressing hours of waking experience into these tiny, impossibly fast bursts of neural activity. And the slow oscillations will catch those ripples and carry them upward, toward the neocortex, where they'll be woven into your longer-term memory networks. All of this will happen while you're not paying attention, which is, honestly, kind of the point. But let's stay here for a little while longer, because there's something worth talking about. Something that brings together so many of the threads we've been following tonight. And it has to do with where all of this is heading. Not just the science of brain oscillations, But the way our understanding of these rhythms is beginning to reshape how we think about consciousness itself. Here's the thing. For most of the history of neuroscience, consciousness was considered almost untouchable as a scientific question. It was philosophy. It was the hard problem, the question of why subjective experience exists at all, why there's something it feels like to be you. And for a long time, most serious neuroscientists steered clear of it, because there didn't seem to be any way to get traction on it experimentally. You could study perception, you could study memory, you could study motor control, but consciousness? That was a career risk. You'd end up sounding like a philosopher, and in many neuroscience departments, that was not a compliment. But Oscillations changed that conversation. Or at least, they gave people a way in. It started, really, with the work we talked about earlier, Wolfsinger and Charles Gray's discovery that neurons responding to the same visual object synchronize their firing in the gamma band, around 40 hertz. That was about binding, about how the brain takes distributed pieces of information and links them into a unified percept. But some researchers started asking a bigger question. What if that binding process, that oscillatory synchronization, isn't just how the brain represents objects? What if it's how the brain generates conscious experience? One of the people who pushed this idea furthest was Giulio Tononi, an Italian neuroscientist working at the University of Wisconsin-Madison. Tononi had trained with Gerald Edelman, a Nobel laureate who was deeply interested in consciousness, and by the early 2000s, Tononi was developing something he called Integrated Information Theory, or IIT. The core idea is deceptively simple. Consciousness corresponds to a system's ability to integrate information. The more a system can combine diverse inputs into a unified whole, the more its parts are both differentiated and interconnected, the more conscious it is. Tononi even proposed a mathematical quantity for this, which he called phi, using the Greek letter. A system with high phi has lots of integrated information. A system with zero phi, like a simple thermostat, has none. Now here's where oscillations come in. When you're awake and conscious, your brain's oscillatory activity is complex, varied, rich, with different frequencies interacting across different regions. There's gamma nested in theta. There's alpha modulating attention. There's beta coordinating motor plans. It's this incredibly intricate, ever-shifting symphony of rhythms. And critically, these rhythms are integrated. They talk to each other. They're coupled across regions. The brain, in its conscious state, is a system with very high informational complexity. But when you lose consciousness, whether through deep sleep, general anesthesia, or a severe brain injury, something very specific happens to the oscillatory landscape. It doesn't just slow down, it loses complexity. Marcello Massimini, a colleague of Tononi's, developed a remarkable technique to measure this. Starting around 2005, Massimini used transcranial magnetic stimulation, a brief magnetic pulse delivered to the scalp that activates a patch of cortex, combined with high-density EEG, to watch how that pulse of activity spread through the brain. In a conscious person, the pulse triggers a complex cascade of responses, rippling outward through multiple regions in an intricate, differentiated pattern. But in deep non-REM sleep, or under anesthesia, the same pulse produces a simple, stereotyped response, a big wave that either stays local or spreads uniformly, like dropping a stone into thick mud instead of a clear pond. The complexity collapses. Massimini and Tononi turned this into a measure they called the Perturbational Complexity Index, or PCI. And here's what's genuinely remarkable about it. PCI can distinguish between conscious and unconscious states with extraordinary accuracy. It works in patients under different types of anesthesia. It works in different sleep stages. And most importantly, it works in patients with disorders of consciousness, people in comas or in vegetative states, or in that heartbreaking gray zone called minimally conscious state, where someone might have flickers of awareness but can't communicate. PCI has identified patients who were clinically diagnosed as vegetative, unresponsive, apparently unconscious, but whose brains showed complexity levels consistent with consciousness. Some of those patients, when later tested with other methods, turned out to be aware. They were locked in, conscious, but unable to signal it. Think about what that means. An oscillation-based measure, derived from the way brain rhythms respond to a perturbation, can detect consciousness in someone who shows no outward signs of it. That's not a small thing. That's potentially one of the most important diagnostic advances in neurology, and it's built entirely on the foundation of understanding brain oscillations on everything from Berger's alpha rhythm to steroids' thalamocortical loops to Singer's gamma synchrony. And this connects to something else that's been quietly revolutionizing how we think about anesthesia. For most of the history of surgery, anesthesiologists monitored depth of anesthesia using crude measures: heart rate, blood pressure, whether the patient moved. The terrifying reality is that anesthetic awareness, where a patient is paralyzed by muscle relaxants but conscious during surgery, happens more often than most people realize. Estimates vary, but something like 1 or 2 in every 1,000 surgical procedures may involve some degree of awareness. That's tens of thousands of people every year, worldwide, who are awake and feeling pain, but unable to move or cry out. EEG-based monitoring has been used in operating rooms since the late 1990s, with devices like the bispectral index monitor, or BIS, which processes frontal EEG signals into a single number from 0 to 100. Below 60, you're probably adequately anesthetized. Above that, you might not be. But BIS has limitations. It can be fooled by certain anesthetic agents, and it doesn't capture the full complexity of what the brain is doing. Newer approaches, informed by the kind of oscillatory science we've been talking about, are much more sophisticated. Emory Brown, an anesthesiologist and neuroscientist at MIT and Massachusetts General Hospital, has done extraordinary work showing that different anesthetic drugs produce distinct oscillatory signatures. Propofol, for instance, produces a very characteristic pattern of frontal alpha oscillations coupled with slow oscillations, a signature that looks remarkably like an amplified version of natural sleep spindles. Ketamine produces something completely different, a high-frequency gamma pattern that reflects its dissociative mechanism. By reading these oscillatory signatures, anesthesiologists can potentially titrate drug doses with much greater precision, keeping patients in exactly the right state, unconscious enough for surgery, but not so deep that recovery is prolonged. And Brown has made a point that I think is worth sitting with for a moment. He's argued that general anesthesia is, in many ways, the most dramatic everyday demonstration that consciousness depends on oscillatory dynamics. Every time someone goes under anesthesia, their consciousness is switched off, not by destroying neurons, not by removing brain tissue, but by altering the patterns of oscillatory activity. Change the rhythms, and you change the state of consciousness. Restore the rhythms, and consciousness returns. It's as close to a proof of concept as you can get that oscillations aren't just correlates of brain function. They're constitutive of it. They're part of the mechanism. So where does all of this leave us? Nearly 100 years after Berger's first recordings, brain oscillations have gone from a curiosity to a cornerstone. They're central to how we understand sleep, memory, attention, perception, consciousness, and disease. They're therapeutic targets. We can boost them, suppress them, entrain them. They're diagnostic tools. We can read them to detect seizures, assess consciousness, monitor anesthesia, and possibly catch the earliest signs of neurodegeneration. And they're still, in many ways, mysterious. We understand the mechanisms of many individual rhythms. We know about T-type calcium channels and the reticular nucleus and thalamocortical loops and inhibitory interneurons. But the full picture, the way all these rhythms interact across the whole brain to produce something as seamless as your experience of being alive, that's still being worked out. It might take another century. It might take longer. But here's what I find most comforting about all of this, especially right now, as you're lying here and your brain is doing its nightly work. The oscillations don't need you to understand them in order to function. They've been refined by hundreds of millions of years of evolution. The slow oscillation that's beginning to roll through your cortex right now is essentially the same rhythm that rolls through the cortex of every sleeping mammal on Earth. Mice, cats, dolphins, elephants. It's ancient. It's conserved. It works. And there's something almost poetic about the fact that the deepest, most restorative phase of sleep, the phase where your slow oscillations are biggest and most synchronized, where your spindles are most abundant, where your hippocampal ripples are most precisely timed, is also the phase where you are least aware of anything. The brain does its most important maintenance work in the dark when you're not watching. Memory consolidation, synaptic homeostasis, metabolic waste clearance through the glymphatic system, all of it happens in the deep quiet of non-REM sleep, orchestrated by oscillations that you will never consciously experience. So let them do their work. You've spent this time learning about what your brain does while you sleep, and now it's time to let it actually do those things. The alpha rhythm has probably faded by now. The theta is here, slow and wandering, carrying whatever last fragments of thought are drifting through your mind. Soon the spindles will come, those elegant little bursts from the thalamus, and they'll help close the gate between you and the outside world. Sounds will become distant. The feeling of the pillow, the weight of the blanket. These will fade from awareness as the thalamus stops relaying sensory information to the cortex. And then the slow oscillations will take over, deep and steady, pulling everything into their rhythm. Your cortex will pulse with them, millions of neurons switching on and off together in a pattern so fundamental it predates the evolution of primates, predates mammals, may even predate the split between the lineages that led to birds and reptiles. You're participating in something genuinely ancient tonight, something your brain shares with creatures that lived 100 million years ago. Hans Berger would have loved to know all of this. He spent his career convinced that the brain's electrical rhythms held the key to understanding the mind, and he was right in ways he could never have imagined. He didn't live to see sleep spindles mapped, or gamma synchrony discovered, or sharp wave ripples linked to memory, or oscillations used to detect hidden consciousness in unresponsive patients. But every one of those discoveries traces back, in some way, to that stubbornly persistent man in Gina who believed the waves meant something. They do mean something. They mean everything. And right now, they're carrying you into sleep, gently, the way they were always designed to. So let them. Let the slow waves come. Let the spindles do their work. Let the ripples replay whatever needs replaying. You don't need to hold on to any of this. Your brain will keep what matters. That's what it does. That's what the oscillations are for. Good night.